Good afternoon, everybody. Nobody's in yet. Don't expect anyone, but I'm going to go ahead and record it. So tomorrow, one of the things I want you guys to do, if you're in my live class, is bring your notes with you to labs so that we can review for your exam. Today, we are doing chapter 23, digestion, so we'll go ahead and get started. Okay. This is an image directly from your textbook that shows the different parts of your stomach. This is not the beginning of your digestive tract, but it's one of the most significant ones. One of the things that most people don't realize is not a lot of digestion occurs in the stomach. Its main job is to store the food, to sterilize it, and dose it a little bit at a time to the small intestine, which does most of the digestion and absorption. Okay, this is another image directly from your text. It shows you what the innermost layer of the stomach looks like, all the different cells. And then if you dive into one of these indentations here, gastric pits, they're called officially, you see these different cells here that are going to be producing acids, mucus, bicarbonate buffer, things like that. Okay, so we're starting at the beginning, the oral cavity, which is your mouth, side of mastication, which is chewing and swallowing. Your salivary glands produce saliva containing salivary amylase. Now, what is salivary amylase all about? Well, it's really important for a couple of reasons. First is it helps you realize or understand that starch contains sugar, because what it does is it breaks down starch. Amylase is an enzyme, ASC ending, that breaks down amylose, which is starch. And what that does is allows you to know that starch has sugar in it, so it's a source of energy. Secondly, it helps wash starch off your teeth, which loves to stick on there and provide a source of energy for bacteria, which produce acids and eventually give you dental caries, which is cavities. So getting rid of the starch that's on your teeth is really important. That's one of the main jobs of the salivary amylase. The more saliva you have, the more movement you have, the more washing you get of your teeth. And so your saliva keeps your teeth clean, and so does your mouth. The esophagus is connected to the mouth, and it connects the stomach. Really, its only job is to get food from your mouth past your thoracic cavity, which is your chest, down into your stomach. Your stomach, there's several parts to it. The fundus is the upper part that's attached to the esophagus. The body is the main central part. And the rugae on the inside are the folds. Now, this kind of acts like pleats on a skirt or on a whatever something would be folded up. What it does is it allows expansion to happen. So when you put a whole bunch of food in there, it doesn't stretch out the surface. The folds allow it to basically expand out without stretching. This is important to protect the inside of the stomach. The antrum is the lower portion attached to the duodenum, and the pylorus is the very end of the antrum. It contains the pyloric sphincter, which regulates how much chyme, which is what food is called when it arrives in your stomach and mixes with digestive juices, how much chyme is allowed to go from your stomach into your small intestine, begin in earnest the process of digestion. Okay, your small intestine is the site of most digestion and absorption. The first part is called the duodenum. Its main job is to receive all different kinds of fluids from various parts of the digestive tract. So the main thing it's receiving is chyme, which is food mixed with digestive juices and acid from the stomach, but it's also going to be receiving bile, which is coming from the gallbladder, which helps you digest fat, in addition to digestive juices from the pancreas, which are going to help neutralize the acid coming out of the stomach. So the jejunum is the main portion. It's the central part of the small intestine. That's where most digestion and absorption occurs. And the final part is called the ileum. It's attached to the cecum. So when you have a valve between the two, or a joint junction, it's called the ileocecal valve or the ileocecal junction. So when you see that in review, it means it's a junction between the ileum and the cecum, so ileocecal. The pancreas back at the duodenum produces digestive enzymes. That's its digestive function. Remember, it's also going to be producing insulin and glucagon for sugar balance. The liver produces bile for lipid digestion. It moves that bile into the gallbladder. And then when you have a fatty meal that's sensed by the stomach and the duodenum, it releases that bile to help you digest fats. And we'll talk about how that works in a moment. The large intestine is very important. And it's the colon, primarily is the colon, is the removal of water from the feces. So your food, as it's being digested, is very much like soup for most of it, or stew, however you want to think of it. And then when it reaches your large intestine, the job of the large intestine is to remove most of the water. This is super important because if you didn't, you'd have diarrhea all the time, which is a source of giggles, But in the third world, it is not very giggle worthy because people die of it all the time. So the idea is you don't want to lose a lot of water in your feces, and that's what one of the jobs of the large intestine does. In addition, there are bacteria living in there if you're healthy. They produce vitamin K and other vitamins that you need for blood clotting, as well as other important biological functions. The end of the large intestine is called the rectum. It's for compaction and storage of feces until defecation can occur. And then, of course, the end is the anus, the exit of the waste. So now we're to the end. Okay, this is an image from your text. There's the duodenum right here. In this spot is where you would see the stomach attached. Just below it here, you would find the pancreas. And then the jejunum, ileum, and large intestine are shown here. This is a great image. I like it. Okay, so this shows you how motility works, or the movement of chyme through the digestive tract. So in peristalsis, which is the top one, and by the way, this is directly from your text, the food moves by squeezing a ring of muscle right behind it, sliding that ring down. So it's kind of like trying to move something through a tube, something solid inside a tube with your hand. You slide your hand down. But in this case, it's not an object moving down. It's a ring of contraction that slides down. This would be smooth muscle contraction. So this is how food is pushed through. Now, segmentation is a little bit different because it's not really designed to move the food as it is to mix the food. So imagine if you had two substances in a bag. How would you mix those two things if the bag was full? You'd have to just kind of squeeze various areas. By squeezing the different areas, you would mix it inside the bag. So that's called segmentation. Okay, chemical decomposition or breakdown of food molecules. Simple carbohydrate foods are sugars. Complex carbohydrate foods can include both fiber and starch. The difference is you can digest starch, you can't digest fiber. Enzyme hydrolysis is the chemical process of breaking down large molecules into smaller molecules using water. And if you've ever studied this, what it does is water breaks into two pieces. Each of the pieces connects to the piece of the material exiting or splitting, allowing it to stay separate. Amylase is an enzyme that digests starches into smaller disaccharide or two sugar subunits. Maltase digests maltose into glucose. So maltose is made of two glucose molecules. And so maltase breaks that down into individual glucose molecules, which can then be absorbed. And then sucrase, something we study a lot in our labs, our general biology labs. They digest sucrose into two molecules, one of glucose and one of fructose. Lactase is something that some people are missing. It digests lactose, which is milk sugar, into glucose and galactose. And if you're missing that, and that lactose ends up in your large intestine, it makes gas and indigestion, and that's lactose intolerance. Okay, so let's take a look at this. This is glycogen, a starch made up of a whole bunch of monosaccharides or sugars. So when you take amylase, you break it down into these different disaccharides. Maltose made of two glucoses, sucrose made of one glucose and one fructose, and then lactose or milk sugar into that, just like I said. Okay, I didn't want that. Here we go. Proteins are composed of chains of amino acids, and they're called polypeptides because each of the bonds that connects amino acids to each other is called a peptide bond, as you can see right up here. So each protein has two ends. The amino terminal end is named that because this is an amino group, NH2, and this is the carboxyl terminal end because this molecule here is carboxylic acid. So when you take an endopeptidase, which breaks up it in the middle, so you're basically splitting it into two larger pieces. So those are endopeptidase because endo means an inside, not on the. So exopeptidases do the opposite. They cleave the distal or terminal amino acid off. So aminopeptidases cleave off the amino end, and carboxypeptidases, as you can imagine, cleave off the carboxyl terminal amino acid, giving you individual amino acids and peptides. Now, please note, whole proteins can be absorbed by the small intestine and often do. And this is what leads to certain food allergies. We'll be talking about that later. I don't know why it's doing that. Sorry about that. Folks, fats are composed of two parts, 3 fatty acids and 1 glycerol molecule most commonly. Most fats are hydrophobic and will not mix with water. So, they don't, oil and water don't mix. It's because of the fact that the fats are non-polar and water is very polar. So bile emulsifies fat into small water-soluble fat droplets. What does emulsification mean? Well, instead of having a big blob of fat, it breaks it down into lots and lots of tiny ones. What's the significance of that? Well, having lots of tiny bubbles of fat increases the surface area. And that surface area then is going to be attacked by lipases, which break down fats and allows you to break it into the fatty acids and glycerol so you can absorb. Now, most of us don't need a whole lot of fat to survive, but our ancestors definitely did because they were always concerned about starvation. So being able to digest fat and absorb it was important if you were going to get the calories you needed to survive. Okay, so triglycerides are broken down into monoglycerides and free fatty acids. I'll show you that. So there's the triglyceride. There's the monoglyceride, which is the glycerol and a single fatty acid and 2 free fatty acids. So this is what's broken down into, and then your body absorbs these. Okay, this is not a figure from your text, but this one from a previous text that I really like. This is a terrific overview if you want to learn how the digestive system works. So what is it do? What is it doing? So M stands for motility. How is it being moved? S for secretion. What things are being secreted into the lumen of this area to help with digestion? D, what digestion occurs there? And A, what absorption occurs there? So this is probably your best single image for studying what happens in each of the main regions in terms of motility, secretion, digestion, and absorption. I highly recommend you study this to get the basics of how the digestive system works. Okay, this is a figure directly from your text showing you basically the same thing, not quite as good, but if you combine the two figures together, it does give you an excellent overview of what's happening in the different parts of the digestive tract. Okay, the cephalic phase of digestion. Now, what does cephalic mean? It means your head. So it's inside your head, your brain. And so your brain knows that food is going to be coming, especially when you're hungry, when you smell food, when you think about food. So it's the anticipation of food. So GI basically stands for gastrointestinal. Motility increases. That means your stomach starts to move. And because there's gas in it starts making squeaking and rumbling sounds, and that's that rumbling sound that you hear when you're hungry. So basically your stomach is saying, hey, I'm ready for food. Go ahead and put some food. GI secretions begin before food arrives. So the idea is that when the food first arrives in your stomach, there's already digestive enzymes there so that the digestive process gets a kickstart. Saliva is produced in anticipation, especially when accompanied by smell. So when you think about food and smell food, you start to salivate. And who was the researcher? Oh gosh, it slips my mind. I'll remember. He was a Russian, Pavlov. Pavlov did tests with dogs where he would ring a bell right before he fed them. And then that trained them to think, okay, when the bell happens, food's coming next. And he would put these little cheek tubes and he could measure how much saliva was being produced by the dogs. And what he would do is he'd ring the bell and they would start producing saliva in anticipation of the food. We do the same thing, not with bells usually, or cheek tubes, but we do have dinner bells that we ring, and that tells people, hey, food's coming. All right, your mouth is the first part of your digestive tract. Chewing or mastication starts the digestive process here. Grinding food breaks up the food to increase surface area for digestion. If you swallow a big chunk of food, it's much more difficult to digest. That's why mom always said, chew your food. You mix the food with saliva that allows for swallowing and to begin the digestive process. A little bit of digestion occurs, especially starch, as I mentioned earlier. What does saliva have in it? Well, water is for liquefaction to make anything solid like saltines into a liquid or kind of a slurry. Enzymes, which help begin the digestion. Mucus allows it to slide down your esophagus, especially if you're not drinking anything. Lysozyme is an enzyme that kills bacteria. And immunoglobulins, which are also bactericidal. The idea is you trying to, by the time the food leaves the stomach, the food's supposed to be completely sterile. That's one of the main jobs of the acid in your stomach, is not to digest the food. There's a little bit of digestion that occurs that way, but mostly just to kill the bacteria in there so that you don't end up with a bacterial infection in your small intestine. The food's supposed to be sterile, not always, especially if you have a big meal, but that's the main idea of these materials as well as the acid in your stomach. Okay, swallowing, scientific term is deglutition. You guys have heard of gluttony. It comes from the root for swallowing or eating, glut. So deglutition is swallowing. Pressure against the uvula begins the reflex. When you push food to the back of the mouth, that little dangly thing called uvula, when that gets pushed backward, that initiates the non-conscious or subconscious, unconscious reflex of swallowing. The glottis rises and the epiglottis covers the windpipe. So the glottis is the top of your windpipe. The epiglottis covers it. So when the glottis rises up, the epiglottis covers it. That way you don't get food or liquid into your lungs and cough. The upper esophageal sphincter opens. Basically, you're opening the esophagus to allow the food to go down because otherwise it's sealed off to keep the food down in the stomach. The lower esophageal sphincter opens. That's the one just above the stomach. Now, what is heartburn? Well, basically what happens is when you pack the stomach too full of food, imagine trying to stir a bag that's completely packed with material. If you've packed it so much, you can't really fit anymore, and then you zip it up. Well, if you try to mix it, what would happen is little bits would squirt out of the seam, out of the zipper. And so that's what's happening. Because your stomach is so full of food, when it starts to squeeze to mix the food in your stomach, some of it squirts up into your esophagus. Why does it burn? Because your esophagus doesn't have that layer of bicarbonate buffer and mucus that protects your stomach. That's not there. So when that acidic chyme comes up into your esophagus, it burns. I used to get that a lot when I was eating really big meals. I don't eat big meals anymore, so I don't have that reflux problem. So more frequent, smaller meals will reduce the probability of gastric reflux and heartburn. And it's called heartburn because that area is right behind your heart, so it feels like your heart is burning. All right, the main functions of the stomach are storage and sterilization of food prior to digestion. So not a lot of digestion happens in your stomach, so get it through your mind that the main job of the stomach is to hold the food and sterilize the food and dose a little bit along the day to your small intestine. So what are the secretions? I call them excretions. They're still secretions because they're coming from basically inside your body to the outside. Hydrochloric acid, HCl, lowers the pH, which rises the acidity level. It sterilizes the food and softens it. Now, the softening has to do with denaturing the proteins, causes the proteins, which are kind of like this, to unfold open. That allows enzymes to get in and break those peptide bonds. Lipase is 10% of all digestion occurs in the stomach. So these are lipases breaking down fats in your stomach. Pepsin is a unique enzyme in that it doesn't end with the ASE suffix. But pepsin is interesting because it won't work in neutral solution. It starts off as pepsinogen, which is the precursor molecule. When it hits acid, it becomes pepsin. Now it can start to break down proteins. Now, why do you start breaking down proteins in your stomach? Well, proteins are difficult to digest. They have to be denatured and unfolded. Meat is difficult to digest. So starting in the stomach is a great way. And as it says here, it's activated by hydrochloric acid. If you put pepsin in with meat and in a neutral environment, it won't work very well. It only works in acid. Protection of the walls, bicarbonate buffer is found under the mucus. These two layers help protect your stomach from the acid and enzymes that are breaking down the food in your stomach. Lumen. Okay, so how is hydrochloric acid produced in your stomach, but not in the cells that made it? The way that it works is the hydrogen ions and the chlorine ions are moved separately, so that the hydrochloric acid itself only exists in the lumen of the stomach, and yet through a variety of kind of shell game exchanging ions, bicarbonate buffer moves out, and you can see the potassium and hydrogen and chlorine move out, and then there's a variety of chemical reactions. And here I'm not going to ask you all the detailed chemical stuff on there, but it does show you how hydrochloric acid is produced in the stomach. Okay, so this shows you the physical way it's set up. The stomach lumen is very, very acidic. There's a layer of mucus, which is the physical protection. And then right under the mucus is the bicarbonate barrier that neutralizes the acid. And so right there at the surface of the cells, it's neutral, even though the center of your stomach is very, very acidic. Okay, stomach ulcers. So the least common is the one that most people believe is the most common. It's called Zollinger-Ellison syndrome, and that is too much acid produced. So most people believe that the reason why people get ulcers is they produce too much acid, and they believe then that's due to stress. Not true completely, but in these cases, when way too much acid is produced, something like Tagamet or Zantac is an over-the-counter drug that can be used to reduce stomach acid production. Now, if this excess stomach acid is allowed to go on for a long time, it can actually tear a hole in your stomach wall, and that allows that material in your stomach to go out into your abdominal cavity and can lead to all kinds of problems, including death. The most common, believe it or not, are two things: aspirin and bacteria. You might not have thought of it. Well, why aspirin? Well, aspirin is actually a chemical called acetyl salicylic acid. It is an acid. Now, normally when we put it in our stomach, it stays in with our food and it doesn't press up against the wall of our stomach, but it is an acid. And when you consume aspirin, it's going to make your stomach slightly more acidic. And if it presses up against the wall, it could potentially start to dissolve your stomach, believe it or not. And that's why people take aspirin that's buffered, as in buffering. It's got a buffer included with it, so it doesn't produce acid when you consume aspirin. Most common actually is a bacterial infection. It's called Helicobacter pylori. Pylori is the Latin for stomach. Remember we talked about the pylorus was the end of the stomach. So these bacteria live under the mucus layer and actually destroy the stomach lining. So this is the most common reason for ulcers, not excess acid production. So keep that in mind. What are some treatments? In the past, antacids. Now, what's the problem with antacids? Well, you produce a bunch of acid, right? But then you neutralize it. The problem is you end up with a thing called alkalosis. And that is you're destroying hydrogen ions by absorbing it by another chemical. And alkalosis means your whole body becomes alkaline. And that's a problem. Having slightly alkaline body is normal, but an exceptionally alkaline body can actually cause all kinds of problems. So antacids are really not the best solution for excess acid production. Most commonly today are drugs which block acid production, such as Tagamet and Zantac. Most people don't really need it, but if the doctor does diagnose you with Zollinger-Ellison syndrome, you'll get one of these, and which are now, I believe, over the counter. So I gave you the examples. All right. No, that's not what I wanted. Okay, intestinal phase. Secretin is a hormone that's released when acidic chyme enters the duodenum. So basically it says to your body, hey, there's acidic chyme coming from the stomach and the small intestine. We need to start doing something. So what it does is it creates a negative feedback loop, which means that it inhibits acid formation in the stomach. And the idea is that if you're sensing acidic chyme coming in, you don't need more acid. So it's basically a way to slow down the production of acid. It also slows gastric emptying rate, another negative feedback mechanism, meaning, hey, we have plenty of food coming from the stomach and the small intestine. You can slow down now. And it also stimulates the pancreas to produce the bicarbonate buffer, which is going to neutralize the stomach acid, because the job of the acid was to kill the bacteria, unfold the protein, begin the digestive process. But now in the small intestine, those enzymes work at neutral or slightly basic conditions. So the bicarbonate buffer has to make the food now neutral or slightly basic in order for those enzymes to work. Okay, another hormone is called cholecystokinin, or CCK. It's released when chyme-containing fats enters the duodenum. So the idea is it's a hormone that says, hey, there's fat coming in. We need to start doing something to help digest the fat. What it does is another negative feedback mechanism, inhibits acid formation and slows emptying, the same way secretin did. But the key to CCK is it stimulates the gallbladder to release bile. So bile comes in, it breaks up the fat from large chunks into small ones, and that allows the enzymes to get in there and digest it. Now, you don't want fats ending up in your large intestine because bacteria will get a hold of it. They'll produce gas and acids and all kinds of problems, and you can have indigestion and diarrhea if large amounts of fats enter your large intestine. Okay, intestinal phase. In the duodenum, digestive enzymes for all food types are found. Bicarbonate buffer neutralizes the acid that's produced by the pancreas. Into the blood, well, what are we talking about? Intestinal phase. I think we're talking about the pancreas here. It's kind of behind my little thing here. Anyway, so these are the two hormones that are produced. We already talked about that earlier. Why does the blood go from the digestive tract to the liver before moving to the heart? Well, the way that that works is here's the blood going from your abdominal aorta through your digestive tract. It's going to be picking up food as well as giving up oxygen. Now, instead of the blood going directly back to the heart, which is the normal way, it goes to the liver. Now, why does the liver take it first? Two basic reasons, a main reason and a minor reason. The minor reason is to identify and detoxify certain substances that are coming out of the GI tract. For example, if you absorb something you're not supposed to, like alcohol, your liver has a shot to detoxify it. But most importantly, there's a whole lot of food molecules that are going to be coming through here. What your liver does is store much of them, especially sugar, in it so that your blood sugar levels don't rise so fast right after a meal. And then between meals, the liver releases that sugar so that you can maintain your blood sugar during the day without having to eat or even be digesting. For example, if you haven't eaten in a couple of days, the food's gone completely from your digestive tract, but you can maintain blood sugar levels by your liver releasing sugar from the glycogen that it's stored, which is a starch in the liver. So the idea is you send it to the liver so it can take out excess food and get rid of toxic materials before it gets into the general circulation. That's the answer there. Okay, this is an image of the cross-section of the liver. Bile is produced in liver in these things called hepatocytes. Each of these cells is called a hepatocyte. It literally means liver cells. Bile moves through the common hepatic duct, which you can see right here, to the gallbladder, makes a little U-turn and goes up into the gallbladder. This happens most of the time, but then when you have CCK and fat in your stomach, the gallbladder squeezes, it squirts down into, all the way through this back pathway here, and right into this duodenal papilla, which is a little bump here, and it starts mixing with the food coming from your stomach so you can digest the fats. Bile moves down through the common bile duct, which is right here. When fatty chyme, which is food mixed with digestive enzymes, enters the duodenum. Bile emulsifies fat in the food, makes them water soluble so that the enzymes can get to it and break it down. Okay, fat digestion. This shows, it's doing it again. It's showing you how this happens. Now, this is not going to be an essay on your test, but it shows you how large blobs of fat, when mixed with bile salts, form these little tiny particles. And then these little particles go into even smaller ones called micelles, which are microscopically small. Now, in this case, the enzymes can get to them and they start breaking them into monoglycerides and free fatty acids. Then it goes into the cells of the intestine, goes through the smooth endoplasmic reticulum. If you remember your general biology, smooth ER is there for packaging and processing lipids. So it takes care of those lipids and puts it into little bubbles, which then go to the Golgi apparatus. The Golgi apparatus puts it in a thing called a chylomicron, which is going to dump it into the, not the blood, but it makes a little detour here into the lymphatic system and eventually to your heart. Now, they're not exactly sure why this is the case, but I think what it is to avoid having to move fatty particles into a high pressure system. If you put it in the vena cava, it's a much lower pressure situation and just better all around. Is that it? It's just a picture? Okay. All right. Carbohydrate digestion in the intestines. Amylase from the pancreas hydrolyzes. That means using water to break down from large particles to small. Complex carbohydrates into simple sugars. Brush border enzymes are enzymes that are found right on the surface of these cells. They convert disaccharides into monosaccharides. So what's the point of putting an enzyme right on the surface of the inside of the intestine? Well, when you break it down right on the surface, it can go immediately in. Because think about it, if there's digested food out in the middle of your small intestine, it doesn't do you any good. It's got to get to the intestinal mucosa and get into these cells if you're going to get it. So by having it digested right on the surface, it's a terrific way to kind of kill two birds with one stone. It is digest it and then absorb it right in the same spot. So absorption, how do you get molecules, some of these molecules from your lumen into your cells? Well, there's a thing called a sodium glucose symport active transport mechanism. So in this case, you actually use energy to move glucose from the lumen of the intestine into the cells. But in fructose, it's facilitated diffusion, and facilitated diffusion does not require any ATP energy, so therefore passive. Eventually, it goes to the extracellular fluid, which is this tan area, then eventually to the capillary, again through facilitated diffusion using proteins that help move these particles from the intestinal mucosal cells through the ECF and eventually blood. No, don't do that. Okay, protein digestion. So I talked about this a little bit earlier, but I'm going to go through it in a little bit more detail. Large proteins are hydrolyzed or broken down using water, pieces of water, by peptidases, which are enzymes that break down peptides, proteins, into dipeptides, which are two, peptides, which are large ones, and amino acids, which are individual ones. Okay, so notice that there's different ways that you can absorb this. I'm going to get to the picture here before I finish the rest of these notes. Di and tripeptides can be co-transported with hydrogen ions, so you can end up with di or tripeptides. You can end up moving individual amino acids, but here's the significant one. Small peptides are carried intact across the cell by transcytosis. That means a bubble forms around it and pulls it in, and then eventually dumps it into your blood. So the question is, why would you want to do that? There's a couple of reasons for that. Well, when you're a baby, this is how you get antibodies out of your mom's milk that you've been consuming into your blood to help protect you against bacteria, viruses, and other microbes. Secondly, it also helps your body identify any proteins that are coming in as potential microscopic invaders, so it can start preparing an immune reaction to it. So this is the reason why whole or partially whole proteins are brought in. And by the way, the downside of that is that this can cause food allergies. Because of the fact that your body does pull in these full proteins, please note that proteins from strawberries, from peanuts, from shellfish are made of the exact same amino acids as all other foods. It's just the fact that they're connected together in a certain order gives rise to the food allergy. And that would not be the case if you broke down every single food into individual amino acids. You would never get a food allergy, but because you bring whole proteins through, that ends up in the blood, your body attacks it and overreacts, and that can lead to anaphylaxis and death eventually for people who are sensitive or sensitive to those molecules. All right, so you can use sodium and hydrogen co-transport mechanism to move these particles in, eventually goes to the extracellular fluid, and eventually the capillary with blood. Large proteins may be absorbed using endocytosis, which can cause food allergies. And this is how newborns receive antibodies in colostrum. It's also the way your immune system prepares for invading microorganisms and causes food allergies. I don't know why it's doing that. Okay, the intestinal phase, water absorption. 9 liters of fluid into the small intestine per day. 9 liters, that's 4.5 2-liter sodas. That's way more than your body even has total liquid. So what it means is it's producing a bunch of water, it's going through your digestive tract, and then it's recycling it back out in the large intestine, putting it back in it. So it's like a big circulating system. 9 liters of fluid into the small intestine per day. It absorbs about 7.5 liters of water per day. So that means that most of the water that's going into your food to help you digest it gets absorbed back in the small intestine. But now 1.5 liters of chyme move into the large intestine. This is a pretty big bottle of soda. That's still diuretic or diarrhea. So your body's not done with it. So when the chyme moves from the small intestine to the large intestine, it stops being called chyme and starts being called waste. There you go. The large intestine reabsorbs about 1.4 liters per day. That means 0.1 liters is all that leaves with your feces normally. It excretes a small amount for lubrication. So a little bit of water is left in the colon so that the poop can get out. All right, diarrhea causes dehydration by disruption of water absorption mechanisms. So your body's trying to reabsorb the water, and the bacteria usually are producing these substances that prevent that from happening. So why would bacteria want to prevent you from reabsorbing water? Why do they want you to have diarrhea? Well, it turns out that if you have diarrhea and there's bacteria in there, they can enter the water stream much better than if they were in a stool, in a solid poop. So by entering the water supply as diarrhea, it makes it much easier for them then to get into another person. I know it sounds nasty. In places where waste and drinking water are not separated very well, that's how people get sick over and over again. So the bacteria have learned over millennia that by giving people diarrhea, it allows them to spread to other people faster, and that's what they're trying to do. So as an osmotic diarrhea, water is held in the intestinal lumen by solids such as lactose. Remember we talked about lactose intolerance, and olestra is a fat substitute that was popular years ago. In secretory diarrhea, excess excretion of water by the large intestine due to bacterial toxins. The best example of that is cholera. So cholera bacteria produce these toxins which cause you to not be able to reabsorb the water. You have a lot of diarrhea, and the bacteria are trying to get back into the water supply and into other people. Hey, this is an image directly from your text. It shows you the different regions of the colon. The cecum is the very beginning of it. There's your variform appendix or appendix. And then the rectum is the end of it. The sigmoid colon is called that because sigmoid means S-shape. So you can see kind of an S shape here. And then the anal canal leads to the anus. Okay, bacterial fermentation. So bacterial fermentation does occur in your large intestines, and this is good. And what normal flora in your large intestine do is they produce vitamin K. Vitamin K is necessary for the clotting of blood. So if you don't have healthy bacteria in your large intestine, clotting blood can be more difficult. So bacteria in your large intestine are good if they are healthy. Now, if you put certain food molecules into the large intestine and don't fully digest them, you're going to generate gases. So for example, people who are lactose intolerant, the lactate, which is lactic acid from undigested lactose ends up in there. The bacteria get a hold of it and they start producing acids and gas, can cause lactose intolerance, diarrhea, gas, etc. And then beans, you guys all know what happens when you eat beans. The proteins are difficult to digest for many people. Those proteins end up in the large intestine. The bacteria break it down, release methane as a byproduct, and out comes the gas. Okay, digestive health, protection, and problems. So immune defense. So M cells in your large intestine and small intestine engulf antigens from the intestinal lumen. Remember we talked about how large antigens made of protein usually are absorbed, and that's basically how they identify them as being a foreign invader. So they basically absorb it and say, hey, look what I found. Guys, get ready. This stuff is going to be coming in potentially as a whole virus or bacterium. Macrophages, which are white blood cells, and lymphocytes, also white blood cells, then receive those antigens from the M cells and can initiate an immune response. So your body's ready for it as soon as those bacteria or viruses enter your blood. In irritable bowel syndrome, or sometimes called irritable bowel disease, it's chronic inflammation of the gut due to normal GIT contents. Now, one of the review questions and test questions says, is irritable bowel disease or irritable bowel syndrome due to an unusual and highly toxic material? And it's no. The answer is normal gut contents that your body's just reacting to in an inappropriate way. And the scientific name for vomiting or throwing up is emesis. It's due to contraction of stomach muscles and reverse peristalsis. Remember, peristalsis is supposed to be pushing the food down. Reverse peristalsis pushes it up. And so when it's normally what's happening is that your body's saying, hey, there's stuff in our stomach usually, and our upper part of our small intestine sometimes, that we need to get out because there's a problem. There's toxins in there, usually, that we want to get rid of so you don't absorb it. So in the chemicals in the blood, certain chemicals like alcohol, certain levels of pain can cause that to happen, or vertigo, basically motion sickness, seasickness, car sickness can be. Now, one of the problems with throwing up or vomiting too much is it leads to alkalosis again. Remember, when you get rid of too many hydrogen ions, you start becoming alkaline, and that can be a problem. The other problem with vomiting too much, especially for people who vomit regularly, which are bulimics, it can lead to esophageal ulcers because you're sending acid through the esophagus over and over again. Remember, the esophagus doesn't have the protections the stomach has. And tooth decay. Why tooth decay? Because you're putting acid on your teeth. Your teeth are basically very highly modified rocks. Rocks dissolve in acid, so you're going to lose a lot of your teeth if you vomit a lot. Okay, that is it for today. Nobody showed, which I didn't expect anyone to, but hopefully this presentation helped you. I hope to see you if you are taking the 18-week class. I hope to see you in lab tomorrow. We'll be talking a little bit more about the exam. If you're taking this as a 9-week class, just check your Start Here pages and get through your review questions and send me an email if you have any questions. Have a good day, guys. Bye-bye.